Hello, good morning or good afternoon, everyone, depending on where you're joining us from, and welcome to today's webinar. My name is Eleni Tokali, and I am a drugs intelligence analyst at Global Data. It is a pleasure to have you with us today to discover new technologies to accelerate ADC design. Today's webinar is brought to you by Turbine, the biological simulation company using AI to build a digital lab for predictive computational models of human cells and tissues. It is co-hosted by Accenture, a leading global professional services company who provides solutions for accelerated and streamlined drug development. Today's speakers include Dr. Daniel Perez, Chief Scientific Officer and co-founder of Turbine. As a medical doctor specializing in cancer research, Daniel helps design innovative and lasting cancer treatments by translating Turbine's computational findings into clinical insights. His primary responsibility is to understand complex scientific and medical challenges, guiding biologists, translational scientists, and data scientists in generating new discoveries that drive more effective treatments to patients. Daniel holds a PhD and has extensive experience in system biology. His years at Turbine have deepened his expertise in building accurate models of complex biological processes, such as the payload response of antibody drug conjugates. We also welcome Dr. Hassan Nasseri. Hassan leads R&D in next-generation computing for Accenture Innovation. He manages projects in quantum computing, high-performance computing, scientific modeling, and simulations and networking. Hassan joined Accenture in 2018, where his first role was leading an AI team that delivered innovative projects in computer vision, network optimization, and AI-based automation. Prior to Accenture, Hassan worked as a scientist in academia, building novel algorithms for sensor networks and mobile devices using AI optimization and simulation techniques. He received his PhD in signal processing technology in 2018. I'd like to welcome you to our webinar platform. This webinar is browser-based, so if you disconnect, click the link you received via email to rejoin the session. You can ask questions using the Q&A widget or by using the Ask a Question box. And we will try and get through as many of your questions as we can once the presentations have concluded. Please use the Help widget if you experience any technical difficulties or require any more assistance. And please use the related content box to access any additional resources. Without further ado, let's jump into our topic for today by starting with a quick overview of today's ADC market and the challenges that pharmaceutical companies are facing in development. Let's start by looking at the impressive growth of the ADC market over the past decade first. So in 2014, ADC sales were just $1.1 billion. And by 2013, this figure has grown nearly tenfold to $10 billion, with a compound annual growth rate of 27.8%, which is driven by increasing adoption and innovation in this space. Looking ahead, as you can see in the slide, the forecast for ADC continues to be promising with significant growth. So as you can see, according to Global Data's Sales and Forecast database, by 2030, global ADC sales are projected to reach almost $60 billion. This exponential growth is being fueled by advancements in linker payload technologies and improved targeting precision, which enable more effective therapies. However, this success is not without challenges, which we'll explore in the next slides. So while the ADC market is thriving, it's important to highlight a critical dynamic in development progress. Despite a robust pipeline of ADCs, most are still in early stages, particularly in preclinical and discovery phases, which account for the majority of the pipeline. So this heavy focus on early stage development reflects the market's immaturity and many candidates yet to progress further. Meanwhile, the clinical pipeline is expanding, particularly in phase 1 and 2, which make up the bulk of the trials. And this reflects a growing interest and investment in early stage clinical testing. Yet, as you can see on the slide, only 17 ADCs have reached the market stage to date, which is a stark contrast to the high volume entering the pipeline. Additionally, we see that 6.9% of ADC candidates have been discontinued. That's the 110 drugs to be exact. So this gap between early stage activity and successful commercialization underscores the significant hurdles in translating ADC development into approved therapies. So what's driving this disparity? Let's look at some of the key challenges here in ADC development that have led to the current gaps in clinical success. So some of the key challenges here are payload selection and sensitivity, targeting precision and antibody linker design, regulatory hurdles, toxicity, and cost and accessibility. So one of the most critical challenges in selecting the right selecting the right payloads or combinations for ADCs. So payloads interact with cells in complex ways, and finding the ideal combination is not straightforward. So it's crucial to strike the right balance to ensure maximum therapeutic effect while minimizing unwanted effects. Secondly, achieving precise targeting to the disease tissues is another major hurdle. ADCs rely on selecting the right antibody to direct the payloads specifically to the tumor cells and correct linker design. So if the antibody isn't well chosen or the linker design is flawed, the ADC may miss its target or affect healthy tissues, leading to off-target effects. So this takes us to another challenge, which is toxicity. Proper engineering of ADCs is really important to avoid causing significant toxicity in healthy tissues, which then limits its therapeutic potential. So to summarize, in an ideal ADC development process, understanding how payloads and their combinations will affect disease cells is the first step. Once we have identified the ideal payloads or combinations for a specific patient cohort, the next step is designing the molecule itself. This means selecting the right antibody or multiple antibodies and optimizing the linker to ensure that ADCs are targeted precisely and that payloads are released as intended, limiting toxicity. So Turbine and Accenture bring advanced technologies that offer solutions to these crucial steps, helping to accelerate ADC development and improving targeting precision, and ultimately enhancing the efficacy of these therapies. So Daniel, since one of the key challenges that we identified at Global Data concerns payload response, can you elaborate a little bit on the industry's problems and how Turbine is working towards facing those challenges?
Thank you, Eleni, and welcome everyone on this webinar. I'm very happy to give you an overview of what we are building at Turbine, and especially how we apply this technology to answer questions around ADCs, starting with payload resistance. So first, I will talk a bit about what's the vision behind Turbine and what do we build, and then I will transition towards payload resistance and showing a case study about how we tackle these challenges. So basically, we set out to build Turbine with the purpose to have guiding experimentation with simulations before you actually run the wet lab experiments. In nowadays R&D, it's getting more and more complicated to figure out which hypothesis to progress to validation, and you need to build confidence in those hypotheses to really invest in validation. So how Turbine helps with this process is, on one hand, that you are able to generate a funnel which is wide enough and contains good ideas, good hypotheses around what is a good target, a biomarker combination, and you can do this on scale, running patient-relevant avatars in silico and look for this hypothesis. But as I said earlier, it's not enough to get a good hypothesis. You actually need to build that confidence that you would like to bring that forward and validate it. So for that, on one hand, we are focusing on giving more mechanistic interpretation and understanding of the hypothesis, which really helps to build this confidence and basically justify the investment in the downstream analysis. And secondly, it's a fast process, so we can be much faster with simulations, even compared to knowledge graph-based approach or other currently standard data science tools. So you have the chance to iterate more quickly. And all of these, in the end, leads to a wider and more efficient funnel. how do we do this? So we have built the simulated cell around the notion to have one single representation of human biology, which can be used across many different cell types, many different indications. Today, we are focusing on oncology and building patient representative avatars of cell lines, organoids, PDXs, patient samples, to then be able to run these in computational simulations, ask new questions, translate those hypotheses, and then run the validation and learn from it and do this again and again. This is the turbine learning group, which is represented on this slide. The first step of this process is to train these avatars, which means that we are using similar network-based representation of the different cell types, using their omics data sets to parameterize these and basically building a good representation of these different biological samples. And then we train them based on their experimental behavior, being that in an in vitro setting or being that the patient responds. When we have good trained and benchmarked set of avatars, which can be hundreds or thousands of different heterogeneous set of models, we are able to run the novel simulations where we are introducing alterations which the system has never seen. This can be monotherapy-like alterations, this can be combinations, it can be like a drug modifier screen. So there is a variety of different tools you can build around these avatars and basically run these in silico simulations. One of the most important steps after this is to understand which are the interesting ones you would like to bring to validation. So on one hand, we use the outputs of the simulations to translate these results, but also we complement that with cutting-edge computational biology toolkit to really give a mechanistically explained set of hypotheses, which then can be fuel validation. The next step is to validate these hypotheses, which can be done in our own in-house laboratory or with partners, where we are really focusing on validating the hypotheses coming from the simulation. So you can actually select the models, the assays, the perturbations based on what the simulation told you. So it's on one hand quicker to figure out what's the validation plan, but also we see that there is an increased chance of likelihood for success. And the final step, which can be also in the beginning, is when you actually miss data to train these avatars and simulate, there is a need to generate data fit for purpose for that question, what you are asking. That's also something which we can do in-house or with partners. So there is no step in the process where you would say there is not enough data. That level of data which is needed for turbine simulations can be generated throughout the process. It's not that high amount which is not feasible to generate. But okay, how do we leverage this technology to help ADC design? So on one hand, I would like to emphasize that what we are focusing on today in Turbine is pretty much related on resistance mechanisms and really understanding the biology, mostly around payload response. But if we zoom out a bit, then we can classify these resistance mechanisms into these four different categories. So the first is antigen-driven, where you are focusing on how alterations in antigen expression can drive resistance, which we classify to intrinsic or acquired. So for example, a lower level of antigen expression in response to the ADC treatment can lower the efficacy, or a mutation in the antigen can also demolish this efficacy throughout time. What we are more interested in nowadays, and recent ADC research also shows that most of the resistance is most likely going through payload response, is how payload-driven intrinsic and acquired resistance can be better understood. So what we are focusing on there is, on one hand, really modeling the internal working of the cells to help guiding, for example, how DNA damage repair or cell cycle and apoptosis pathway are driving this resistance, and what are the potential markers which can be tracked to avoid intrinsic resistance or to tackle acquired resistance with combinations. And the other is how a payload response can be changed throughout time through altered metabolism, efflux mechanisms, or mutations in the payload targets. So overall, from these different questions you can ask from the platform, we basically have three different solutions, which I will show in the case studies. So the first is you can actually match payloads with responsive cancer subtypes. So there is a possibility to run many different cancer indications and their subtypes and test the different payloads in these indications and pick the best one for your question. So if you are looking for NSCLC subtype and you would like to have the most efficacy in that subtype, then we can pick which payload can deliver that efficacy. The second is predicting biomarkers for patient selection. So even when you have a good fit of payload and patient population, you should avoid certain patients, or you should actually include certain patients, so you can both design inclusion and exclusion criteria. And finally, to tackle especially acquired resistance, designing rational payload combinations is something which is more and more important as we progress towards clinical use. So let's dive into a case study where I can show what does this mean in practice. So in this case study, we have been focusing on the question of why patients are not responding in a certain subtype of NSCLC, specifically squamous cell carcinoma, to topo-1 payloads. So this is a question which is coming up in recent trials, and we are very interested in helping to answer this question by better understanding NSCLC biology, and inside that how SCC and other subtypes are different to each other, and why do we see this potential difference. One reason can be the high genomic instability and different molecular drivers in SCC, but it can be also related to the target and how topoisomerase 1 interacts with this biology, and what is the DNA damage repair connection of topo-1 inhibition and the SCC biology. So what we have done to tackle this question is, first I'm focusing on one payload, which is SN38, the topoisomerase 1 inhibitor payload. And what we have done is we simulated, we first trained the avatars and then simulated the response to this payload across many different cell types representing these different indications. So what we were interested in is, first, if we can see an overall correlation between the in vitro IC50 and the in silico IC50. And as you can see there is a very good Pearson correlation between the two responses. So you can state that the model could capture the response, and this is a test set, so these are unseen cell lines where we have seen this nice correlation. Then the second more interesting question for us, if we can actually delineate the response patterns across the different subtypes. So here what you see is an IC50 distribution across the different subtypes. Here are the labels of the subtypes. So for example, the purple one is the squamous cell carcinoma, and here you see the in vitro and the in silico distributions. And what you can tell from this graph is that there is a very similar distribution of response across these subtypes in vitro and in silico, which tells us that the model could actually capture the subtype-specific effects as well, beside the general cytotoxicity pattern. Okay, so this is a good start. And then the next question is, if we can capture this response, do we actually understand what is happening behind the scenes? So if you zoom into that, we ran a biomarker screen where we actually can perturb the different nodes in the system and see which ones are changing the response to the payload. We highlight here one example which came out from the platform without training on this, and then we have found a lot of evidence that this could be actually a very interesting driver of resistance, namely SLFN11. So what you see in the simulations in this case is that you have a control cell where you don't have SLFN11 perturbation, and you have a certain distribution of response. And then you introduce this alteration in silico. So this is not an in vitro knockout, this is an in silico experiment, and see how that changes IC50. And we can see that in almost all the cases, it causes resistance, so the sensitivity is decreased. What we can also see on the individual cell line level, so this is one specific NSCLC cell line, is that compared to the normal dose response in the simulations, once you add the knockout of SLFN11, you actually cause resistance. You can't reach more than like 80% inhibition in the cells. So on one hand, with that, you find a potentially interesting modulator of drug response. And on the next part, we see that if you dive into the model and try to understand why this actually happens, you can very nicely build a representation of the mechanism. So what we see here is, on one hand, we have the topoisomerase inhibitor here, and on the other hand, we have the SLFN11 knockout on the other side of the chart. And basically, you can track down the signaling through stalled fork, RPA pathway, and in the end, it will cause break-induced replication, which will lead in the end to cell death. So basically, by looking at the model and going back to the graph representation of signaling, which I mentioned when we construct the simulated cell, you can basically build a mechanistic understanding of why a certain biomarker can potentially mediate respond to a certain payload. So from this, we build this mechanistic hypothesis that schlafen can potentially mediate the response to topoisomerase inhibitors, specifically SN38 in this case. And when we looked into the literature, we have found that this is actually something which is now tracked, at least in preclinical models, but also potentially later in clinical trials. And indeed, this could be a potentially interesting mechanistically connected marker of topoisomerase 1 response. So this shows an example of what can be done to understand payload response and predict biomarkers which are potentially relevant. The second example is once you have these resistant models in silico, then you can start tackling the resistance by combining different drugs to each other. It can be combination of payloads, or it can be combination of small molecule inhibitors and payloads. So in this case, what we have done is we have been looking at first a big in vitro screen, which was recently published, the GDSC squared, where there are many drug combinations and synergy metrics for a lot of drugs, which are including some payloads. And one payload was luckily SN38, for which there was some combination data which we could use. Synergy is a rare event, so we could focus on olaparib combination where actually synergy happens in vitro. And this chart shows how nicely the platform could predict the combination synergy between SN38 topoisomerase 1 inhibitor and olaparib PARP inhibitor without seeing this combination data and training on the combination data. So it's very important to highlight that this result is just training on monotherapy data and then predicting combination synergy. And that you can see that the correlation between the in vitro response and the in silico response is around 0.8 Pearson correlation, which is a very strong correlation. So this highlights that with the platform, just knowing the monotherapy data, which is usually available for payloads, targeted therapies, etc., we can, with high certainty, predict combination synergies, which then can be brought forward to in vitro validation. And you can also build an understanding, similarly to schlafen around why that synergy occurs. So to summarize these findings, basically what we have seen is that, of course, in the ADC field, the biggest question is therapeutic window and how you can manage that therapeutic window. And you can break it down to two main categories. The first is to increase the likelihood of response, so basically increase efficacy. And on the other hand, how you can decrease toxicity. And in an ideal world, we could increase efficacy while decreasing toxicity, and you have a wider therapeutic window, so more patients will benefit longer from that certain therapy. So what Turbine can help in this era is, on one hand, help designing better certification strategies to increase likelihood of response by first focusing on payloads, but we are moving forward also modeling the whole ADC as a conjugate and not just the payload itself. So the technology evolves to give you more and more deep insights on the biology. Secondly, we can have designing combinations of, let's say, dual payloads or cytotoxic drugs combined with small molecule inhibitors. We also have the capability to help figuring out if there is an immune component, an immune relationship between, let's say, payload response in this case, and can that guide ADC response? And finally, you can also use a platform to identify novel payloads or even potentially novel targets. And on the other side of the table, where we have toxicity questions, we are not that useful at this point, but luckily we have Accenture colleagues here who will help you to talk about mostly about linker chemistry and how that can improve tolerability. Thank you very much.
Thank you. So I will continue. Hi, everyone. As already Daniel introduced you to the topic of payload design, which is typically a toxin that will target the cell, we will continue with the linker design, which is another component of the ADC. So at this point in the call, probably all of you know what is ADC, but just to remind is that we have a linker which connects the payload to the antibody. The linker needs to have certain properties, which is basically also the molecule that in most cases is the main design thing that we do. Of course, we also need to engineer the antibody, but that comes more of a naturally. And payload, as we discussed about it already, and the linker is the part that has a lot of flexibility of the design, and it needs to serve two main purposes. One purpose is that it needs to be able to carry, so I will talk about this later a little bit, but it needs to be able to carry basically the payload together with the antibody, and also it should be cleavable to release the payload. And there should be certain other properties. But before going to that, I just want to bring this up from the beginning of this call about ADCs, that there are, I have written here 13. Already we heard about 14. Okay, correct number could be 14. 14 approved ADCs in the market, but we need to know that this research in ADCs started more than 40 years ago. So this shows that it is a very difficult field of research. So we need to speed this up. Another aspect of this is the cost. Average development cost of an ADC nowadays is around $5 billion. And the cost of treatment is upwards of 200K. This is average annual cost of treatment. Many public health organizations or public health basically systems cannot even afford some of the new ADCs. So the goal is to reduce the cost. How much? Maybe by a factor of 1,000. So it's not a twofold or a tenfold increment. You may need to reduce this by a factor of 1,000. And so there's a lot to be done in ADC design. There are also other targets that we need to look at to reduce the toxicity, to improve the efficiency, and so on and so forth. The idea is that in silico methods will help us with that. So let's look at one component, which is the linker. As I said, there is an attachment group in the linker which attaches to the antibody. We need this group to be stable enough so that antibody can carry the payload through the system, through the body, and deliver it. And the other side is the cleavable. We are looking at the cleavable antibodies. In this case, when antibody-drug conjugate ADC goes into the cell, then it gets cleaved, so the payload gets separated. There are other forms of design as well, but here we are looking at the cleavable one. In this case, we need a site in the linker that when ADC is inside the cell, an enzyme can break it. So those are the key components of a linker being a linker. But then there are few other properties that you may also want to consider. You want it not to be toxic to the body, you want a certain solubility, and so on. So there are many others. And also you need it to be synthesizable, manufacturable. You need to be able also to conjugate them and create the whole structure, and so on and so forth. So in the design of a linker, which is similar approach to many small molecule design, again, we are designing a big biologics drug here, but the linker itself is a single molecule. We are starting with an initial data set, which tells us what are the possibilities. And that data set we created from all the public available data on ADCs and linkers. We collected and created that data set. Then you need to generate your molecules, and then you need to react and combine and create your ADC. You also need your protein structure. Once you have done these steps, you have a 3D structure or the full model of the ADC with which you can go to do your further simulations. You can do molecular dynamics, you can do reaction kinetics, or you can do more higher level and more larger models as turbine is producing. So I will quickly go through the steps of this, but before going that, this is a demo that we created. It's not an asset, it's a sort of a prototype that we created at Accenture using latest AI tools to showcase how AI can be used and why we are using a certain tooling to build this. So let me come back to this overview and tell you the tooling that we are using. Here we are using mostly state-of-the-art AI tools, and also our focus is on using microservices, containerized solutions that can be easily scaled up. So the idea is that we can provide computational advantage by using tools that are more efficient. We can improve the usability so that end users, R&D researchers can easily use them, integrate them. We can improve the accuracy by training and fine-tuning these models, which are done, for example, companies like NVIDIA and others are fine-tuning and creating the models that are highly accurate. And then also the idea of accelerated design is here as well, which that if we are building a use case, let's say for, in this specific case of ADC linker design, then that works as a template, a blueprint for an R&D to adopt. So the first step of it, as I told about the database, is that we have a database of common linkers. That database needs to be created. We already created a database, but more things added there would help. Then you pick an initial linker from that database. It's a database of things that have already tried and proven to work. We look at the properties of that. In this case, we look at, for example, a log-P synthesizability score. It is 1. Let's say our goal is to improve the synthesizability score. The linkers that we have in our database, the good ones, are difficult to synthesize and scale up. So with that goal, we need to have a molecule generation process. And in this molecule generation process, we are using, in this case, we are using a regression transformer model. We are using also recently not even released GenMol from NVIDIA. So these models start with an initial scaffold or initial structure that you give and generate many similar molecules. And the generated molecules are weighted and scored based on these properties that you like. For example, you want to combine with cysteine using a maleimide group. Okay, you specify that. And then we filter for that active binding group. Then you say that, okay, I want this to be peptide-based, and synthesizability score I'm targeting should be larger than 1.5. So we score these, filter them, go through the iterative loop, and generate more and more new ones. So here is a short video of showing you iterative steps of generating new molecule. In this case, in each step, 10 new molecules are generated. The best ones are picked, feedback to the model, and saying that, okay, this was good, generate a similar ones. After that, we get, for example, a data set of 100 new linker molecules, which are generated based on a structure that we asked them. They have that linking point, they have the both attachment group and the cleavable group. They have the desired properties. Then we go further. We want to create the full ADC and see, is it, first of all, realizable chemically? Is it working in silico? And then go further with the simulations. So you pick a toxin here. As already discussed, it's in itself, it's a research process to design the toxin. But let's say we have a database of those toxins which are, or payloads basically, which are created. And from that database, you pick one. In this case, we select this one as a payload. And then we need to go forward. And so there's a short video showing you that we can do it. We can create a 3D structure of the payload as well from the smiles or from the description of the molecule. The 3D structure requires doing some energy minimization so that we find the most stable conformer of the molecule that we want. So we need then to do the reaction to combine. The first stage, which we are doing here in silico, by the way, in practice, this will be done in a manufacturing process. But we are doing the reaction in silico to study it's feasible or whatnot. So we specify this is the linker, this is the payload, do the conjugation of the linker plus payload. We do that, and we create that structure. Next step in the process would be for us to look at the antibody itself. We need to get the 3D structure of the antibody. There are two ways to get it. One is that we can do protein folding. There are models like AlphaFold or ESMFold, which can help us to do the folding of a protein if we don't know. But in this case, there is also a protein database that we can get the structure from that if it's a known antibody, which in most cases for the ADC design, the antibodies are well-known structures. Then you need to just modify the structure a little bit. People will do some genetic modifications of the cell line to produce an antibody with certain characteristics that you can do your linking, or basically you can do your bonding with the payload. So in this case, we specified the PDB token, it loads it, and then we need to prepare that. For preparation of this molecule, we need to create an environment around it, which is in this case basically aqueous environment. It's in water, so we need to put the hydrogens, or basically the protons around it, protonate, to adjust for the acidity, for the pH. We set it for 7.4, for example, as a blood plasma. So we set certain properties. We tell that what is the linking site, which is the cysteine, and what is the docking area, the region that the cysteine is in that. And some preparations needs to be done. So those preparations are done, and a grid is created around that site. So this box that you see here, inside the box, a docking grid gets generated. So in this grid, again, I'm not going to the details of how the docking will be done or how the payload linker will be combined, but to suffice, the box is discretized, and all the points that docking can be done, all the energies are there calculated. Next step, we need to do the docking, or basically combine the payload plus linker with the antibody. I'm showing these steps one by one, not saying that how specifically this is done, but what I'm trying to tell here is that these are done all automatically. You see, in silico, without any manual intervention. The way that this is done is that we identify how the linking will be done, how the bond, the covalent bond in this case, will be. Since this is cysteine, we know that what is the part of the cysteine that will bond. It can be automatically fetched from, again, a database, that there is a sulfur carbon, there is certain structure. We create that structure and attach it to the linker. So we basically attach part of the antibody side chain to the linker and then overlap it. So we go and overlap those atoms, and then the covalent bonding is done. So there is a search for the overlap. In this case, there's only one carbon sulfur shown, but in practice, it's a longer chain. You need maybe 4 or 5 atoms in that side chain. Once you specify that, then we do the overlapping of those, and we create different poses. So in this case, there is a short video showing you that how different poses of the, okay, this is the antibody, this is a payload and linker with the side chain attached, and the next video shows different poses that generated when we do the covalent bonding. So the attachment group, the covalent bond is there, but different poses are possible. So we find all possible poses or realistic poses, and out of all those poses, you can pick the minimum energy one. The minimum energy one will be the best pose. You pick that, and then you have your 3D structure of the ADC. So there's a video here as well to show you this example of the minimum energy one that we picked, and you get your 3D structure. With this 3D structure, which is still not the fully optimized structure, you need to go from this to further modeling. The proper step is to do then some energy minimization, either with DFT or molecular dynamics, to get a better pose. But we didn't continue with that. With molecular dynamics, now you can easily continue from here with molecular dynamics to study the stability of this, how stable is the overall ADC in different environment. And it can be also the cleaving or the release of the drug also can be studied, but it will require a more complex simulation setup to study how the enzyme will react and will cleave. So those can be studied with molecular dynamics. Again, since you have the full 3D structure, you can go ahead from molecular dynamics, you can go one step further, and you can study the reaction kinetics and understand how efficient are these reactions, what are the reaction rates that will happen, and so on and so forth. So the goal of this demo so far was to show you that up to this step of creating a full 3D structure of an ADC in silico can be done fully automatically by combining the technologies that are available today in the market. From here, from this step going forward, also can be done with a little bit of work you can continue with automating the molecular dynamics and even some reaction or kinetics or pharmacokinetics, which is not done here but can be done. So the goal of this experiment was to find a linker that has a better synthesizability, and in this case this linker has synthesizability score of 2, and all the other properties are specified here. The idea is that we started from initial linker, which was not very easy to synthesize. We created a linker that will have a shorter retrosynthesis pathway or easier production, which is one of the key challenges in ADCs, actually the efficiency or the yield of production that contributes to a lot of cost related to ADCs. With that, I conclude my demo part or my presentation. Thank you.
Thank you so much, Daniel and Hassan, for those presentations and for providing an overview of the solutions that both Turbine and Accenture are working on to accelerate ADC drug development. Just a reminder for the audience to ask any questions, please send them via the Q&A box at the bottom of your screen. But we already have some questions that have come in. So let's start with the first question. Can Turbine's platform cover new payloads beyond DXd?
Yes, that's a good question. So I was focusing on this actual case study now. But on one hand, all the currently clinically used and preclinically used payloads, being those microtubule inhibitors, topoisomerase inhibitors, can be modeled. There are some novel ones where there is less data in the public domain. So there we are generating some proprietary data in-house, so we can actually have a better modeling predictivity for those. But generally, yes, if the data is missing, we can generate that. And we are also looking into the new fields of combining with DDR inhibitor payloads or potentially immunomodulators as well. So generally, different payloads can be captured, even combinations of payloads with the model.
Thank you very much. I'm just going to go on to the next question. Can Turbine model multiple combined payloads of an ADC?
Yeah, so if the short answer is yes, as I already alluded to that during the demo, but if we are thinking about how that can be done technically, that's not really what we are doing, but in terms of understanding what happens biologically, if you have both payloads in the cell, how those are interacting with the cell biology, that's something what Turbine can simulate. As I have shown for the olaparib SN38 combination, similar synergy readouts, but also mechanism behind the synergy can be captured.
Thank you very much. Going on to one more question. Is antibody or compound antibody and payload effect modeling on Turbine's roadmap?
It is. So not a trivial question, because on one hand, there is, of course, the effect of potentially the antibody, which we don't really understand yet, what the antibody is doing in terms of downstream signaling, which contributes to the efficacy or toxicity. There's the combined nature of modeling the antibody effect and the payload. And there is, of course, the linker, the metabolism, and everything in between. So we take a step-by-step approach, starting with the payloads, combinations of payloads. And the next thing what we are working on is how payload response can, on one hand, modulate antigen expression, as the platform can predict post-treatment transcriptomic state. Basically, you can predict how antigen expression can change under therapeutic pressure, which can be a potentially interesting resistance mechanism. And the other is how you can match patients with certain antigen expression with the right payload. That's the other angle what we are taking. But we have some good early feasibility data that even the whole ADC as a construct used for training can be modeled with relatively good predictivity. We are building the basic understanding around this predictivity now.
Thank you very much. I think we've covered enough for today, and yeah, that's all we've got time for. I think that just leaves me to thank Daniel and Hassan for the great discussion today. To the attendees, you will receive an email tomorrow telling you how you can access the on-demand version of this webinar, or you can access arenainternational.com/events. And please continue to ask questions as the on-demand version remains interactive. And thank you once again, and I hope you will have a really nice day.
Thank you, everybody.
Everyone, bye-bye.
Bye.